All right. So thank you, James, for the last session. We will be doing, so like I've said on the schedule, we would normally have a 45-minute extra session before our 45-minute lunch break, but because of the Zoom issues today, we got off to a little bit of a late start. So we will have a quick 15-minute extra session. So just a reminder that like all other sessions in this workshop, the session is being recorded and live streamed to YouTube and will be available for viewing again in the future. Um, Today, what we'll be doing for our extra session is we will just have three quick lightning talk presentations from CompuCell 3D team members to show some of the research that they've done in the past using CompuCell. And we'll have a few more people presenting tomorrow. Today, we'll have Joel Vanin presenting about his virtual cornea model that he's been working on the last year or two. We'll have Pedro Castel talking about an influenza model that he's worked on. And then we'll have Gilberto Thomas finishing up before lunch talking about his foam coarsening model. So we have a couple of people interested in foam research. Should be interesting to see what he's doing. So I will go ahead and turn the screen share over to Joel if you're ready to go. And I think we have enough time for about 5 minutes for each presentation today.
Yeah, James already introduced a little bit about the work that I'm doing for my PhD. Um, so it's about a virtual cornea. We're being reached out by the people on PNG to create a virtualization of the cornea, as James explained, um, given how exposed to the cornea is. Sometimes if a chemical interacts with your cornea, you can have uh, some damages done that does not lead to a full recovery. These are some of the examples. For example, when you have uh, neovascularization over the cornea, and that impedes your vision. So PNG is interested in that to learn what are the possible combinations of chemicals that can interact with your cornea in a way that is unrecoverable. So to do that, we have to finally understand a little bit of the microbiology of the cornea. That means what are the cell types that are involved in there, how they're distributed. James already gave a little bit of explanation too, that the stem cells for the cornea are usually located on this limbo area, which is the edge of the cornea as a whole, and cells migrate towards the center as the tissue is formed, right? So when we establish the first things that are most important for our simulation, for example, if we're trying to represent a damage and recovery simulation, we first need to understand what are the principles behind the homeostatic state, or there are enough, right, to justify some recovery or some signal for recovery after injury. So in our case, we focus primarily on the epithelium of the cornea at first, but you're going to see a version 2 of this model on the next couple of slides. So I'm here marking where we're focusing first on the epithelium, right? This limbo area, and then this periphery going towards the central part of the cornea. And here are some of the descriptions of what happens in the cornea. It's an interesting tissue because it's like one of the few tissues in our body that has to be transparent so we're able to see. And for that to happen, you cannot have blood vessels, you cannot have certain types of cells in there that would lead to a loss or creating opacity in the cornea. So after we defined the homeostatic state, we can do injury. And here we're following a depth of injury model that says that the time of recovery is dependent on how deep your injury has been. And we can see that after the recovery, if they recover in the similar time that we see reported on papers, to help us to calibrate our model to what is expected. In this example that you're seeing in this video, there is a mild damage being done here because the Bowman's layer was not fully destroyed, but that is enough to activate cells to help reconstruct the basal layer that lead to the proliferation of these cells and the fully recover of the epithelium tissue over here after the injury has been done. You've seen on the right here just a pulse of that chemical, right? To simulate like a droplet. I will replay here so you can see. But there are many ways you can do this in a simulation as a uniform distribute field, not just as a pulse. And in some times when that damage is too deep, the moderate case, you can lead to these adverse outcomes. For example, these ectopic fibrosis, where you have the formation of extracellular matrix in the wrong place, which leads to this malformation that is evident. For example, in case of LASIK surgeries, we have that because on the LASIK, you have to go through the Bowman's membrane, and you can see the cells are not able to recover properly, and you have these patches of opacity in the cornea. So that's the main goal of this simulation, is trying to identify what are the adverse outcomes that can surge from chemical exposure. You said 5 minutes, I used 5 minutes.
Okay, I think I'll jump in already to save us some time and share my presentation. I'll share my screen and jump to here. Okay, so my name is Pedro. I am James' PhD student, and I am developing this influenza model, infection in epithelial lung cells. And I will quickly give a biological background, then I will explain briefly how the model works, and then I will show you the videos. So basically, the virus hijacks the cell machinery to replicate. The cells release cytokines, and the cytokines are sensed by the cells so that they can get to a defensive phenotype. Specifically for influenza viruses, they replicate in the nucleus, and they need to produce proteins in order to replicate before. So there are other types of viruses where the replication occurs before the production of proteins. And they also carry the NS1 protein that is an anti-host protein. It disables the cell defenses. Also, not all viruses are infectious, but they can still produce physiological effects when they get in cells. So my model tries to encapsulate all of these aspects. All right, so my model comprises of an intracellular logic circuit to represent cell phenotypes, genes, and proteins, the spatial representation of the cells, and the field representation of viruses and interferon. Interferon is the cytokine here that I'm most interested in. This is the intracellular network that I built in using MABOS. You will learn about MABOS to model intracellular networks. So each cell in my simulation have one of these. And here I model interferon sensing and how it upregulates intracellular proteins. I should say genes, how it regulates genes, and also viral-related genome segments and proteins, and how they interact with each other. So how the viral sensing system detects viruses, and how the interferon system prevents viruses from replicating. And then this is coupled to the CompuCell processes. So for example, interferon sensing is a process happening in the multicellular system, and this is coupled to the network. And the same happens to endocytosis, virus release, and interferon release. So next slide is on CompuCell3D. This is what I have so far. So I have the cells and the fields, the interferon field and the virus field. With the complexity of my model allows me to perform some experiments in the simulation. For example, knockout experiments. So WT means wild type is when the entire gene of the virus is working, and NS1 del or NS1 deletion is when I specifically deactivate or knock out the NS1 gene. And I can also model other types of situations, for example, high or low MOI, how many cells start out infected. Finally, I will show you guys some videos. So this is an infection case with wild type influenza A in high MOI, and then lots of viral production and very little interferon release. This is with NS1 deletion, NS1 being the anti-host protein. With that disabled, the virus release is much smaller and interferon release is much greater. This is where it gets more interesting when we have a low MOI, so less cells start out infected. Here, 1 in every 100, and infection spreads. This is wild type. Some cells happen to get defensive because not all viruses, newly generated viruses, can come with genomic defects. And finally, with NS1 deletion, quickly leads to plaque arrest because the cell defenses and antiviral defenses are much stronger than the NS1 deleted virus. And I also have some quantitative analysis, but I'm not going into detail on them because it takes some time. So that's it from me. Maybe we should hear from Gilberto now.
Okay, so I am Gilberto Thomas. I am from Brazil, and I work with CompuCell since its origins back more than 20 years ago, when it was decided to write a code like that in Notre Dame, when James was working at Notre Dame. Since then, I have been using this software for a long time. Despite it has been created to biological purpose, it can be used for non-biological purpose. What I'm going to show you is how to use CompuCell to study forms. And basic thing about forms, the first question that appears when you say, why do you study forms? Huh? It seems so silly to work with this kind of things. But I can show here a lot of applications where forms are very important. We have art, food, drink, nature, like a Drosophila wing here. We have a honeycomb, a firefighter using foams to combat fire. We have sneakers with foams everywhere. We have foams in architecture. This is the Beijing pool center during the Olympic Games in 2008, I think. The structure of the building is a structure of a foam, if you can notice that. This particular, you can see my mouse? Now you can?
Yes, yeah.
Okay, see this black stuff here in the vertex of a foam is the use that you can do with foams to extract oil from lichens in the sea, for example. You use foams to remove the oil, and also to mining. You can use flotation in the mine extraction of ore in some So mine is using forms. So forms have a huge amount of applicability. And let's say, but what is a form? Huh? See, you have here three different pictures that some of you may use every day. Mayonnaise, uh, glass, uh, toothpaste, and uh, shave cream. Everything in this picture are kind of forms. Three different kind of forms: emotion, suspension, and basically the more usual forms. The gas bubbles dispersed in a liquid or a solid. You can have a solid form. And the question is, since we have those kind of applications to the phones, we need to understand his stability, how to make a form that can last. And for example, here we have an application, a very new application that you can use a foam to stop bleeding in a gunshot. And it's research from U.S. Army. See, the guy has got a shot here. You put some foam that expands itself inside the body and stops bleeding. Then you can move the guy to the hospital and try to save his life. Okay, but the foam simulation is very simple, very simple. Why does it make it possible to use it with CompuCell? Well, it's the whole characteristic of a form. Its topological structure, the energy minimization, and phase transitions, a lot of structures that make this system, the form system, an excellent candidate to be studied with the POTS model, and hence simulated with CompuCell, which is founded in the POTS model. Okay. What I'm going to show you this time is the coarsening process in the liquid forms. In this process, big bubbles grow expenses the smaller ones. I mean, when you have a foam, for example, in a top of a beer, you see the foam starts to decrease, and some of the decreasing amount, but the number of bubbles decrease because the smaller bubbles gave their gas to the big ones. Okay, this is what I'm going to see here. I can show you here this simulation that I had prepared. It's a very simple simulation, 2D simulation with a grid of 350 square pixels. Each bubble has a 5 by 5 pixel, and this is the target volume, the initial target volume for each bubble in the foam, and lambda volume has some values. I can show you that it's about 3. I start the simulation, and after 100 Monte Carlo steps, I put lambda volume equals to 0. This step is what makes the coarsening process to start, because as James told you before, we have those volume plugin, and the volume plugin is set up with a target volume for the size of the cell, and a lambda volume that say how much the cells must have that target volume. Of course, it can fluctuate around the target volume, but the bigger the lambda volume, the less the fluctuation around this value. If you put lambda volume equals to 0, then there is no restriction anymore to the cell's growth and disappear. That's what Coarsening is. Okay? Let's see if I can show here a small dissimulation. Windows style. See? I put in this simulation a window and you can keep track of the number of the cells or bubbles in the simulation. So you see with time, the number of bubbles are decreasing, but the average volume of the bubbles are increasing. It's a very simple simulation. You can find the simulation in the student materials under the name forms2d.zip. Okay, let's go ahead. Of course, that 2D simulations are not very, very realistic in principle because we live in a 3D world. So I prepare also a 3D simulation just to show you in a very small grid, 50 by 50 by 50. A small simulation because I want to show the simulation working with you, for you. Because the bigger the grid, the huge amount of time you need to run it. So here is the new one. I have two windows open, a 2D cut from the 3D system. Okay, the cut is in the layer 49, but you can change it here if you want. If I put it to run here, after 100 Monte Carlo steps, the lambda volume will be set to 0. And you're going to see the bubbles change the volume here. See the number of bubbles decrease with time in the plot graph here. And you can see the big turning bigger and the small disappear with time. Okay. Finally, I want to show you a more sophisticated simulation. I'm not going to run the simulation because this is a huge one. See, the grid has 200 cube pixels, which means 8 million of pixels in the voxels in the grid. So it takes too much time to run. But I like to show you you can analyze this form with the topological transformation, the phase transitions, that some statistics about the probability of volume size of the bubble, the growth size of each bubble, how many faces each bubble have. I mean, the average number of faces, probability of faces. It's a lot of information you can have from one simulation, and CompuCell can do that. It can treat with big amount of data here. because this grid is very big. You can do even with 300 cube if you want, which means 27 million voxels in the grid. The starting number of the bubbles here is about 40,000 bubbles. So it's a very powerful tool to study this kind of complex systems. This is what I would say to you today. Thank you.
All right. Well, thank you to our presenters. Yeah, our presenters will also be available at the end of the day today if you did want to follow up with them again, but you do need to run for lunch now. So, but if you would like to ask questions now, feel free.
Katrin, go ahead.
I have questions actually for all three of the presenters, if that would be okay. Yeah.
Can you, if you have a question for me regarding the influenza virus, can you do that now? Because I need to head to a breakout room to help another participant.
Okay, so this was a very specific question on interferon responsive factor 1 or 7, I guess 7, which suddenly disappeared from the 4 components after your stud signaling. So it was a dead end. How do you, and after a while it returned when together with the other factors, 3 and 2, I guess, and so on. So how does this go into the network then? Can you build an equation on this, just disappearing factors?
Good question. So yes, the network doesn't connect itself, but it connects to the cells in CompuCell. So I have information in CompuCell in terms of the field. The interferon is a field in CompuCell, and then the network communicates with that field. So when you couple the network with the spatial model in CompuCell, then the network connects itself back.
Yeah, okay. So this is an interconnection between this Modbus or Mobus and CompuCell.
Exactly.
Yeah, I see. Okay. Thank you.
No problem.
So then my next question, or if anybody has a question to Pedro, maybe you want to go first.
Okay. So I think it looks like nobody has a question. So thank you, Katrin. I will
Yeah.
head to the breakout now.
Okay, see you. So then, Gilberto, your foam model, can you predict the stability of the foam? Because this would be really a thing when you think about osteoporosis and all these kind of things where you need a certain stability of a foam-like structure.
Well, stability is a complicated thing because, you know, we have drainage all the time attached to the foam. We cannot get rid of the drainage when you build a foam. Gravity is always there. So since the stability of a foam is how do you say, is promoted by the liquid itself that surrounds that is the liquid matrix which contains the gas. So the stability is obtained by studying this what you call surfactants.
Yeah, but this is in the lung. I'm talking about bone. So tissue.
Bone. I didn't understand. Well, a bone is a solid form, isn't it so?
Yeah, it's more or less a solid foam with a very, very slow remodeling, and therefore the stability is it's stable for a long time, but at a certain number of rebuilding cycles where you get bigger and bigger holes in the bone, you have osteoporosis. And this is a thing for people suffering from, yeah, I don't know the English word, the German word would be Rheuma, and they are treated with a medical, they are medically treated, and this treatment will induce osteoporosis in the neck.
Yes, I heard some about some articles to use the foam to reconstruct the bones, but I don't know how to work with that. There is a research which I just find here, injectable foams repairs bones. But I never work with this kind of solid foam. I'm more the guy to work with liquid foams and bubbly liquids, something like that.
Mm-hmm.
But I know I can, let me share my screen here just to. Can you see here?
Mm-hmm.
See, there is this article here from 10 years ago, which is application of a kind of foam to reconstruct bones. But I never studied this one, to be honest.
Yeah. This, okay. Yeah.
Okay,
So
sorry.
so then my last question goes to Joel. The epithelial cells that you are talking about, when they proliferate, they are not polarized, but in the course of their life, they get polarized and they stop, they cannot migrate any further. Do you think this has something to do with the thing that the operation, the scar of the operation is not repopulated by the epithelial cells, so that they do not make it without the basal membrane to walk to the wound?
Yeah, so there is a strong correlation with the maintenance of proliferation and the ability of the cells to perceive the basement membrane, right? If the cells are not able to deposit the basement membrane for some reason, they will not be able to recover that tissue anymore, right? They lose their proliferative state every time. So then you have a case of recurrent injury where the cells tend to form a closed wound, but after a while, because their proliferation on that area in specific is damaged, is now not able to recover the same way, you see an opening and then a resurgence and then an opening. And that is also shown in the literature. If you're not able to recover your scaffold basically properly,
So your model is you have a hole in the basal membrane, and this prevents the cells to go there and migrate into the hole, so to say, and the hole cannot be closed by the helper cells.
Yeah. So in the case of the cells that are in the stroma, they are the ones that are depositing this new ECM. So the other cells that are the basal cells on top, right? It would be better if I showed you the image. So give me a second. So the basal cells actually can proliferate, but there are some.
But then the defect is not in the epithelial cells, but in the fibroblasts, which produce the stroma.
Yes.
So why are you concentrating on epithelial cells then?
Well, we had to start from somewhere, right? There is no recovery. Yeah. There is no recovery. Let me just share my screen here. There's no recovery of the tissue if we don't have a epithelial cell over there. So we first need to understand how the epithelium works and what are the things that can go astray, and define that very well before we actually go and focus on the damage that can cause something, right? If
So
I don't have
the yellow cells are fibroblasts
fibroblast.
that are going to replace the matrix, right?
Yes.
And then we have this green thing. What is this?
So that's the deposition of the basement membrane, because these cells deposit
Yeah, and
ECM over here.
it's not a real membrane. It's somewhere. It's just thrown in there, and that's why the epithelial cells do not find the right direction, and so
Yeah.
they cannot. Yeah, I see. Okay.
So that happens. This is an example of what happens, right? If the deposition, as you can see, this like strong purple, pink over here, that's parts of the membrane that got stuck in between the epithelium, the same way we see in the simulation. And this was me just running a bunch of simulations. And in one of them, I saw a similar thing happening, right? So eventually one of these fibroblasts walked over here, deposit these in a weird way. And then the other cells just, okay, if I see ECM, I should create my own basement membrane. And that's why we see this happening.
And then polarization of the epithelial cells still occurs, but to the wrong direction. And so the cells just die and the monolayer is not built again.
Yeah. So here I'm not doing polarization explicitly. I'm just saying that if the cell can senses the bottom part, right? If they come detached from it. So I'm not using I'm not saying to the cell, oh, you have an apical and a basal side. I'm just saying if it's more binary than that. It's like if you're touching this or not.
Okay, but this is a plan, of course, to include polarization of the cells.
Yeah. Well, we always have to have in mind what is important for the type of problem we're looking at, because the more information you put it on your models, more complex it gets for you to adjust. And again, a lot of the times we wanted just to do a forecasting on approximation, right? Now, if your study is really focused in understanding what are the actual parameters that are all involved, yeah, then you include more details for sure.
I'm not so familiar with the eye, but from the gut I know you always have stem cells in the gut, and they go to the top of the crypt, and on their way to the top of the crypt, they polarize and they differentiate into real enterocytes. And once they became a real enterocyte, they just die. They drop, they lose their connection to the basal membrane. And this could happen here as well. So if the cell
even on the division, right? If you're describing division like perfectly, this cell would lose contact with it, becomes very columnar, just holding by a thread, and then you have a division. And then the cell that will maintain its format as the basal, it drops again and maintain itself as basal. The other one that lost contact will become the other cell, which is in this case a wing cell. Yeah, so, but sometimes it depends on the scale. For example, each Monte Carlo step, this is something that you're going to learn later, in my simulation is equivalent to 6 minutes. So this whole process that you just described about the clonic, also here on in 6 minutes, that probably happened already, right? Like you have the whole movement and then came back. So for me, it's instantly. I don't have to simulate that. I can just approximate it.
Yeah, very interesting. Really very interesting. Yeah. Thank you for sharing this.
No, yeah, thank you for your comments and questions.
Okay, so we have a lunch break. Not a very long lunch break, but a lunch break. We'll see you back here in 30 minutes. for module 1.3. Thanks, everybody, for participating and looking forward to working with you. Now, the next module will actually start going into the details about how to think about and build these simulations. So we hope that these have whetted your appetite to learn more. See you in a little bit.